Okay, so today we're going to talk about translocation of organic solutes. So we need to be able to define the terms translocation, source and sink, identify where sources and sinks are, and explain why they are those things, and explain how they're translocated, how they're loaded into the phloem vessels or sieve tube elements, and how they're unloaded as well. And look at some of the evidence that used to support their theories. So translocation basically means the movement of organic solutes or compounds from source to sinks via the phloem. So sucrose is the primary one you need to be aware of. So what's a source? So sources are places where organic solutes are produced and loaded into the sieve tubes of the phloem. So if we think about it, your main one you're going to think about is photosynthesis happening in the leaves is going to produce organic compounds, and then they're going to be converted into sucrose and loaded into the phloem. But they're not always the source. If you think about it, in the spring, an oak tree doesn't have any leaves on it at the start of the season, and it has to grow new leaves. So where is the source going to be, and what do you think the sink is? If you think about it, the oak tree needs to make the buds grow new leaves, it needs energy and organic molecules in order to grow those new leaves for cell division. So the roots, actually where starch would have been stored over the previous summer, were being hydrolyzed down into sugars, back into maltose or glucose, converted into sucrose, and then translocated up the phloem towards the leaves. So the actual growing leaves would be the sink here, as they're using the sugars in respiration to release energy for cell division, or using those organic products to make new compounds like proteins and cellulose. So a sink, therefore, is any place where organic solutes are being removed from the phloem, being used by the cells. So they could be being respired to release energy. If you think about it, lots of cells in plant are not photosynthetic, so they're going to need to do respiration to get the energy they need. Just think about in the roots, just the root hair cells doing active transport of minerals. Or they could be being converted into other organic compounds like cellulose or amino acids to make proteins or other sugars and things like that. So for example, making new leaves, as we've already talked about, flowers and fruits, all those sorts of things that are not doing photosynthesis but are requiring resources. And as I mentioned before, they could be being stored away. So we've got energy stores being stored as starch, or maybe in lipids in certain fruits like avocados and things like that. But I think in the roots like taproots like carrots and parsnips, or corms or bulbs like onions, in the roots just as energy storage, and in seeds. So translocation is multi-directional, so it can go up and down, or up or down. It can be going different directions at different times, which is quite unusual. Sucrose is an ideal transport sugar. There have been exam questions about this before. Key thing about sucrose is, remember, it's a non-reducing sugar, so it's less reactive than glucose, which means that fewer reactions are likely to occur with it inside the phloem and inside cells when it's being translocated. It's less likely to be used in respiration. There are special co-transporters that I'll talk to you about that allow it to be loaded and unloaded into and out of the phloem. It's less likely to diffuse out of the sieve tubes because of its structure, and it's a more energy efficient transport system. So the solutes are also moving under pressure, so there's what we call, there is mass flow going on. So we need to think about how is that pressure created, how is the sucrose loaded into the phloem, and how is it unloaded. So there is a model for this which is called the pressure flow hypothesis. I'm going to talk you through that now, and then we'll look at how sucrose is loaded and unloaded as well. So this is a model called the pressure flow hypothesis, a model of how translocation works. So we've got basically some partially permeable membranes here connected up by a tube, and then we've got a sort of bath of water surrounding it. So if you think about region A as being the source, maybe like the leaves where sucrose is being made, and B is representing the phloem, and C is representing the sink where sucrose is being removed and used. And D represents the xylem vessels. So this is a model, remember, it's not how it's actually laid out in a plant. So in the pressure flow hypothesis, the idea is that we're adding sugars, i.e. sucrose, at point A, and that's decreasing the water potential of A. At C, that's the sink, water potential is going to be higher because we're removing sucrose from the inside of C, so it's being taken out and being used by cells. So the water potential here will be higher. So we get a water potential gradient between A and C. Okay, so the key thing here is, though, that's not massively important. What's going to happen is, because of the water potentials, we're going to have movement of water. So at A and C, there's a tendency of water to move in to both because there's a lower water potential in both because there's more sucrose in both. But the key thing is the water potential here is much lower in A than it is at C. So the tendency of water to move in at A is greater than it is at C. So water is going to move in and increase the hydrostatic pressure. Remember, we talked about mass flow earlier. So the hydrostatic pressure is going to be greater in A now because water is moving in than it is at C. So water is going to flow along B towards C, and actually water is going to be forced out at C. And this idea is called the pressure flow hypothesis. And the water is going to return via D. Okay, so what you might want to do now is try and bullet point those ideas without looking back at these screens. Try and do it just from memory and see what you can do. So if you've had a go, the major steps in the theory are: active transport of sucrose into the phloem at the source lowers the water potential of the phloem. Water moves into the phloem at the source by osmosis. This increases the hydrostatic pressure in the phloem, so it's higher than at the sink, which causes mass flow of water with the solutes dissolved in it to flow from the source to the sink. And then at the other part, i.e. the sink, we've got the sugars being removed, and that increases the water potential, so water moves out by osmosis, and also lowers the hydrostatic pressure. So those are your key ideas that are often required in exams. So let's have a look now about how sucrose is loaded and unloaded. So the key thing we've got here is you've got your source, your leaf cell, you've got companion cells, which are often quite specialised and might be highly folded to increase the surface area because they're going to be doing transport across their membranes. The more surface area their membranes have got, the more transport they can do. And remember, your companion cells are connected to the sieve tube elements of the phloem by the plasmodesma. So we've got sucrose is going to be loaded into the companion cells. It's going to move into the phloem, lower the water potential, so water moves in, increasing the hydrostatic pressure at the source. The solutes are going to, or the sap in the phloem is going to flow towards the sink where the sucrose is being taken out and used in these cells at the sink. So, key things: it can pass in by diffusion if there's a loss of sucrose in the source cell, but more likely is that there's going to be active transport using something called a proton pump and co-transporter from outside the cell into the companion cells. Now this uses two specific types of protein. We've got a proton transporter here, which is an active transport protein, and we've got a co-transporter protein here. So we've got a proton pump, which is probably something you've not come across before, where you've got active transport of protons out of the cell, and you've got a co-transporter, which is doing facilitated diffusion effectively, that takes both protons and sucrose into the companion cell. So I'll talk you through that with a little animation. It's a little bit crude, but hopefully it will show you the key ideas. So here's our model. So we've got sieve tube, companion cell. We've got protons inside the companion cell at the moment. We've got proton pump. We've got a co-transporter protein that combines both sucrose and to protons. and we've got sucrose outside in the apoplast root, or also in the cells of the source, and inside the companion cells we've got a higher concentration of sucrose because of previous loading of sucrose. Remember, this is a continuous process, so we have to assume there's already a higher concentration of sucrose inside the companion cells and outside, which means that sucrose is not going to be able to enter by just simple diffusion. It's got to be coupled to some sort of active transport. It also can't move by simple diffusion because, remember, phospholipid is going to stop sucrose from moving, so we have to use some sort of carrier protein anyway. So let's model it through. So the proton pump is going to actively transport protons out of the companion cell and into the matrix, if you like, or the area around the cell, so the apoplast, or the fluid, if you like, surrounding the cells. That means that we're going to end up with a higher concentration of protons building up outside the companion cell than inside. So now we've got a proton gradient between the outside of the cell and the inside of the cell. The protons and the sucrose can both bind to the co-transporter protein. Now there's a proton gradient for the protons to move into the cell, but the sucrose is actually going to be going against its concentration gradient. So the carrier protein, changes shape and carries the sucrose against its concentration gradient and the protons down its concentration gradient into the companion cell. It's basically using the energy built up in the proton gradient to allow the sucrose to be taken in against its concentration gradient. Once it's changed shape, protons are released and the sucrose is also released from the co-transporter, which will change back and pick up more protons and more sucrose, and the sucrose can diffuse into the phloem via the plasmodesma. And the addition of sucrose, as we've already mentioned, lowers the water potential and water moves in by osmosis, creating that higher hydrostatic pressure in the sieve tube element at the source compared to the sink. So the sucrose will flow with the water in solution towards the sink. So where's the evidence that all this is happening? We know that the pressure inside the phloem is really high, 2 megapascals, which is much higher than human arteries, so it's really quite high. It can be really, really fast, much faster than diffusion. That shows us that it's a mass transport, mass flow going on. Microscopy has shown us the adaptations for active transport we've just been talking about. If you poison the mitochondria, that's going to stop aerobic respiration, which means no ATP, and you get no more translocation. So it shows that it's coupled at least in part to an active process. And they've measured the pH of the companion cells and found that it's higher than the surrounding tissue, which is what you'd expect if you were removing protons, because pH, if you remember, is a measure of concentration of protons, and the lower the pH, the higher the concentration of protons. So a high pH means fewer protons inside the companion cell. We can also use aphids, which feed on phloem. Aphids are green fly, and they feed on sugars inside the phloem, and they stick a little proboscis through, they pierce the phloem and extract the sugars from the sap, basically feeding from it. And people have done experiments by chopping the heads off the aphids, just leaving the stylet, the little proboscis in place, and collecting the fluid coming out from the phloem, and you can analyse it and look where the sugars are, especially if you use radioactively labelled carbon in photosynthesis, and see when the radiation appears. It's quite brutal for the green flies, but nice little needles that allow you to extract the sap from the phloem. We know that the pressure in the sink is lower than at the source, and the sucrose concentration is lower at the sink than at the source as well. We still need to answer some questions. We don't know everything that's going on. We don't really understand how it can go at different rates in different places to different sources, and from different sources or to different sinks. So there's other ideas we don't fully understand that you could read more about. Hopefully you found that useful. Thank you very much.